sir.
Good afternoon, everyone, and welcome to BES-Net TT's project webinar. And a special welcome to all persons who are joining us for the first time on this platform. My name is Celeste Chariandi. I'm the Science Communication Officer for BES-Net TT. BES-Net TT is the acronym for the Biodiversity and Ecosystem Services Network Trinidad and Tobago project. And this is a project which is being implemented by the United Nations Development Program and the Ministry of Planning and Development through the Environmental Policy and Planning Division. The project has a focus on pollination and pollinator management and has three components: science, policy, and practice. Under the science component, the project has undertaken three key research activities: a bat survey to investigate bat pollinator species, a seagrass survey to investigate seagrass flowering and pollinators, and a survey of native bees in farmed areas in Trinidad and Tobago. The results of these three research activities are being shared publicly to build awareness of these particular pollinator groups, to underscore their value and status locally, to begin to fill research gaps, and to identify key recommendations for pollinator management. In today's webinar, we will provide a short report on the survey of bat pollinator species, which was conducted about a year ago by a small team led by Mr. Darshan Narang. Before I hand over to Mr. Narang, here are a few housekeeping rules for the webinar. This session will be recorded on Zoom, and the recording will be posted on the BES-Net TT Facebook page and also uploaded to the BES-Net TT YouTube channel. In order to keep our 1 hour time frame, the presentation will take approximately 30 minutes and will be followed by a 15 to 20-minute question and answer segment. While the presentation is being delivered, you may post any questions that may arise in the Q&A area of the Zoom platform. If during the question and answer segment, it is not possible to answer some of your questions, these will be addressed afterwards and will be posted with the recording of the session on the BES-Net TT project page. So as I mentioned, our presenter today is Mr. Darshan Narang. Darshan, you can probably switch on your camera just for a little bit. Darshan Narang is an independent environmental and biodiversity consultant who has been engaged in sustainable development, research, and training projects across the Caribbean for the last 20 years. His background is in protected area management and wildlife ecology, with a passion for birds, bats, and small mammals. Mr. Narang is also a member of the non-governmental organization Trinibats.
Hi Celeste, and hi everyone. Can you please allow me to start my video?
Sure. Darshan, I can share the video from my end.
Oh yes, please. Go right ahead. I was asking you to give me permission to share my camera.
Okay. Okay, we'll do that in a little bit, Darshan, at the end
No
of the
problem.
presentation. Sure.
Go right ahead.
Okay.
Good day, everyone. My name is Darshan Narang, and today I will be talking to you about a survey on bat pollinators in northeastern Trinidad. I belong to a group called Trinibats, along with Shiva Mahadev, Richard Smith, Mary Elise Mango, and Nigel Robinson. Trinibats is a group dedicated towards the conservation and research of bats in Trinidad and Tobago. This project was an initiative of the BES-Net TT project. Today I will talk to you about a summary of the project, an introduction to our project, the method statement that we use to conduct this survey, where our survey was conducted. The two sites were Malau Forest Reserve and the Trinidad Cement Limited quarry site, how we went about collecting our data, and I will then present to you our results and a discussion on the species analysis, including the feeding gills, pollination, and associated floral species. We will then go into the conservation status of bat species found on this project. Bats display a remarkably high degree of diversity in Trinidad and Tobago, and we have approximately 69 species of bats recorded on these islands. For this project, we use BES-Net surveys with the aim of creating a preliminary inventory of bat species present within the survey area. So we spent 48 hours conducting BES-Net surveys at two major sites for this preliminary survey, the Malau Forest Reserve, which represented lesser disturbed primary and secondary forest habitats, and the Trinidad Cement Limited quarry site, which represented a rehabilitated forest. A total of 169 individual bats were caught across 19 species in 3 families for this project. Bat species found throughout Trinidad and Tobago fulfill a wide range of ecological niches and perform a number of vital ecosystem services, such as pollination of important crops, the control of agricultural and disease-carrying arthropod pests, and the dispersal of wild seeds, thus facilitating natural regeneration of native habitats. This project focuses on the ecosystem service of pollination and the roles bats play in rehabilitating disturbed sites. We therefore aim to compare a rehabilitated forest with nearby secondary or primary forest habitats at selected locations to look at the composition of pollinating and seed dispersal gills of bat species found in each site. This comparison will contribute to our knowledge and understanding of bat communities and their relationship to the pollination and seed dispersal in these communities. I will now present to you a brief method statement on the techniques and tools used to conduct this survey. Where did we conduct our study? This project looked at 2 major sites, the Malau Forest Reserve in northeastern Trinidad, representing lesser disturbed forest, and the Trinidad Cement Limited quarry site, the TCL quarry site, which represented rehabilitated forest. So as you can see, both sites on this map relatively close by, just about 5 to 6 kilometers from each site. And as you can see to the left, the TCL quarry site is in the middle of a large conglomerate of quarries, whereas the Malau Forest Reserve is located to the eastern side of the general location. The Malau Forest Reserve was essentially a tall evergreen Mora forest in the past, in which Mora excelsa was dominant. In 1946, Baird reported a continuous canopy layer above the ground with few or no gaps, where Mora commonly accounted for 80 to 95% of the trees in this stratum. Baird also found that the associated species in the Mora forest were the same as those in the surrounding non-Mora forest, and show the same relative abundance to one another. In 1987, a fire devastated much of the remaining mature Mora forest at the Malau Forest Reserve. Successional species were identified and recorded 4 years after the fire in 1991, and the abundances of commercially important regenerating species were determined. In 1998, a survey was conducted that determined there were a total number of species around 201, representing 60 families. There were approximately 14 species of timber value, including Mora excelsa, Terminalia amazonia, etc. This is what the current site looks like at the Malau Forest Reserve. At the Trinidad Cement Limited quarry site, there's a large pond to the southeast of our sample area, and as you can see surrounding this pond, there's a large forest that's been rehabilitated, as the site was previously utilized for mineral extraction in the past. This involved the clearing of the land of all flora, which has now been replanted with the fast-growing pioneer species Acacia mangium. So here you can see our sample site with acacia on either side of the track. So onto our data collection method. Our sampling strategy involved the use of mist nets at ground level to survey bats. So surveys were conducted over a 3-month period for a total of 12 survey nights. 6 nights at the Malau Forest Reserve and 6 nights at the TCL quarry site. Both sites were surveyed in late 2022. The total mistnet effort was around 24 mistnet hours at each site for a total of 48 mistnet hours surveyed for this project. So at each site and each time we set up our mistnets, a temporary field station was set up to process all the bats, as you can see here on the right. That's one of our survey team members, Richard Smith. At each sample site, 3 ground mist nets, 2 of 12-meter nets by 2.7-meter nets, and 1 3-meter long net were erected to capture all flying bats at each survey site. Mist nets were opened from around 10 minutes before sunset to capture those early flying bats, and closed 4 hours after, since this was the time that the majority of bats leave their roost for foraging activities. Here you can see some of the team members setting up a mist net across a cleared survey site within the Malau Forest Reserve. And on the right hand side, there's a pole that's staked into the floor, the forest floor. The ground vegetation is cleared of any debris, and net is stretched across this site. Mist nets were checked every 10 to 15 minutes, and bats were safely extracted within a few minutes and transported in cotton bags to the processing area. All the bats identified to species levels using a field guide from 2015 by Jeffrey Gomes. All morphometric and reproductive data were recorded for each bat. Representative photos were taken of key bat species before being released. Here you can see on the left a bat that's caught in the mist net. You can see it's tangled because they cannot see the nets at night because it's very fine. On the right photo, one of our team members is extracting the bat from the net. And usually we need to do this in the dark, so we have headlights, we have gloves to protect both the bat and our handlers. Here you can see one of our other team members extracting a bat. So Miss Murray Elise is gently removing the mist nets from across the wings. And you can see the bat is being held securely in her left hand within a glove. So this is one method of safely extracting all bats from mist nets. So now I would like to present some of our results and discuss these results in the context of pollination. On the right photo here, this is one of our data sheets, a typical data sheet, maybe not from this project, but it shows you how we collect our data. We record at the top of the form the date, the week, the site. We have the coordinates. If the coordinates are repetitive, we just leave it out. The number of nets that we use per site. And we also have a site description on the right. We describe what flora, what fauna is found in the area, and also other biotic and abiotic factors such as the weather conditions, what the moon, when it is very bright night. If we have tidal conditions at a coastal site, we will record that as well. And within the main content of the form, the data form, we list the number of bats that we caught, what nets, if there were nets at different sites, the time that we caught each bat is recorded, and who handled that bat, and a list of the species of the bat is recorded as well. Usually, if there are many bats, we would put a code. In this case, you see CP, that's for Corolla perspicillata. We record the sex of the bat, whether it's male or female, the age of the bat, the reproductive status, and we can tell these by looking at genitals or presence of breeding in the bat. In order to better get an identification of each bat species, we record the forearm, as that is a definitive feature to tell closely related species apart. On the right-hand columns, we would record the weight of the bats in the bags that we catch them, and then record the weight of the bag, and then in post-processing data, we would calculate the weight of the bat. Also, if there are any special characteristics of the bat that we may see in the field, we'll record that in the comments in the notes section, such as any external parasites, whether there were injuries to the bats, or in this case, I believe bat number 15 was an escaped bat from the net. So we also record bats that escape from the net if we can identify them. For the species analysis in this project, a combined total of 169 individual bats were caught across 19 species. And this is out of the 69 national species we have. So we caught about a third of bat our bat species, just under a third of our bat species at these two sites in this short project. The bats we caught were in three families, the Phylostomidae, Mormupidae, and Emballonuridae. And this was recorded at both Melau Forest Reserve and the TCL quarry site. At the Melau Forest Reserve, we got a total of 103 individual bats across 13 species, and at the TCL quarry site, a total of 65 individual bats were caught across 16 species. For both sites, the two most dominant species were Seber's short-tailed bat, which is also known scientifically as the Corolla perspicillata, with an overall relative abundance of 37%, and the Jamaican fruit bat, Artibius jamaicensis, with an overall relative abundance of 34%. So on the right photo here, we also have one of the characteristics being documented, which is the forearm length of the bat mist-net surveys conducted in 2022. In the table on the left columns, you can see a list of the common names, the species names for each bat species found at both the Melau Forest Reserve as well as the TCL quarry. We also calculated the relative abundance of each species at each site and the overall relative abundance of each species. And here, as I mentioned, we can see the total number of species found was 19, with very similar numbers of species found at both sites. Overall, Melau Forest Reserve showed more individuals caught than the TCL quarry site. This bar chart depicts the overall bat diversity and abundance at Melau Forest Reserve and TCL quarry site. On the left of the chart, you can see the more dominant species, such as Corolla perspicillata and Artibius jamaicensis. And as you go further along to the right side of the chart, these species become rarer and rarer in our survey, such as the Tonaceous saurophylla, Trichopsirosis, and Vampyrolis cartioli, which were only caught once or twice. Let's now talk about the feeding gills, the pollination, and associated flora species for bats surveyed in this project. Species with similar adaptation are assigned to gills or functional groups. Habitat type and foraging mode mainly determine the foraging tasks and thus the adaptations of bats. The pollination of plants by bats is called chiropterophily. Pollination by bats are mainly carried out by the nectarivorous bats and secondarily by the frugivorous bats. Carnivorous bats and insectivorous bats also play a smaller role in pollination and may do so inadvertently when picking insects off a flower or flying through dense forested areas in search of prey. For this survey, frugivorous bats dominated with almost 90% of bats caught at each site, feeding primarily on fruits. Other broad feeding gills identified including nectar-feeding bats, the nectarivores, with two species detected, Geoffroy's tailless bat and Pallas's long-tongued bat. We also caught insectivorous bats, such as the greater sackwing bat, Parnell's mustache bat, little big-eared bat, and striped-headed round-eared bat. We also only caught one individual of a carnivorous bat, the fringe-lipped bat, Trichopsirosis. Here you can see a bar graph showing the distribution of feeding gills of bats at the Melau Forest Reserve and the TCL quarry site in 2022. And as you can see clearly, frugivorous bats dominated both project sites. The two most dominant species were Seabush short-tailed bat, Carolia perspicillata, and the Jamaican fruit bat, Artibius jamaicensis, which are both abundant locally and widespread frugivorous species that play major roles in pollination as well as seed dispersal. Here you can see on the right a photo of the Artibius jamaicensis. Seabush short-tailed bat, Carolia perspicillata, was described in 1961 as a wasteful feeder, but this foraging behavior is beneficial to forest regeneration for disseminating many fruits and seeds. Depending on the number and size of seeds in the fruits, one individual Carolia perspicillata can disperse between 3,000 and 60,000 seeds per night. Two primary nectarivorous bats were recorded during this survey out of a total of 4 national nectarivorous species. Pallas's long-tongued bat, Glossophaga soracina, is often seen at hummingbird feeders left out at night and in similarly forested areas in Trinidad. Though clearly adapted for nectarivory, this bat does not sustain itself entirely on nectar. Instead, its diet varies with food availability, consisting primarily of fruit, pollen, and insects, mainly moths. This species visits and presumably pollinates various flowers, including those of bombaceous and leguminous trees. The seeds of the pioneering shrubs are widely dispersed by this bat. Geoffroy's tailless bat, Anura geoffroyi, the other nectarivore, is known to exploit both mature and modified forests and forage in the understory and along creek corridors. This bat is known to visit and probably pollinate flowers of numerous species. It is also known to disperse the seeds of several species of trees and shrubs. None of the bat species caught in the survey are classified as rare, threatened, or endangered according to the IUCN's Red List for Threatened Species. All species recorded in this survey were currently classified as least concern. Locally, under the Conservation of Wildlife Act, all bat species recorded in this survey are currently protected in Trinidad and Tobago. Here you can see the list of species that we caught on this survey, the IUCN Red List status, as well as the local status of these bats attributed to Jeffrey Gomes' Field Guide of Bats in Trinidad and Tobago. So most of the bat species caught were common to abundant, with a few being classified as rare or uncommon, but this is due to sampling effort over time. If we have more sampling going on, most of these bats will be more common. Here we have a few photos of bats from different feeding gills to show you the facial structures, and this particular bat here is the fringe-lip bat, the only carnivorous bat caught on this survey. So it's a fairly common species in seasonal evergreen and semi-evergreen forests. It's uncommon in agricultural areas and at higher elevations. Small groups roost in caves, road culverts, buildings, and hollow trees. Larger maternity colonies can sometimes be found in deep caves, though this has not been observed yet in Trinidad. This bat takes long flights at early nightfall, hunting on the wing along streams and over ponds, probably in search of its primary prey. What is its primary prey? Frogs. Later in the evening, when frog calling and other activities diminish, the bat switches to perch hunting, employing a sit-and-wait, ambushing foraging strategy as it sallies from a perch in short flights, presumably hunting insect prey. This bat relies on prey-emitted acoustic cues to detect and localize its prey. Though clearly adapted to hunt and consume frogs, this species' foraging behavior appears opportunistic in most of its range, including insects, lizards, smaller species of bats, and even fruit and seeds among its dietary fare. A Neurogeoffroy or Geoffroy tailless bat is an example of a nectarivorous bat. Here you can see on the right a photo of the muzzle being a little bit longer and thinner to reach nectar within flowers. It's uncommon to locally common in lower montane forests, seasonal evergreen, semi-evergreen, and swamp communities and fruit groves. This bat roosts in caves and tunnels. In sealing depressions at Tamana Cave, both sexes hang together in tight clusters, averaging around 15 individuals. They are also known to be found in the Aripo Caves in close proximity to roosting oilbirds, and also at Kumaka Cave in North Trinidad. They are known to exploit both mature and modified forests and forage in the understory and along creek corridors. Diet includes primarily nectar, but they can also take fruit, pollen, and insects depending on availability. The little big-eared bat, or Micronectarus megalotus, is an insectivorous bat, which is rather common and widespread in lower montane rainforests, seasonal evergreen forests, and other forested ecosystems. They roost in hollow trees, log caves, buildings, culverts, and large mammal burrows. This bat is a gleaner that homes in on prey-generated sounds. Diet includes insects, possibly small lizards, and some fruit as well. Insect may be captured by gleaning from the vegetation or in flight. Tilde's yellow-shouldered bat, or Stinera tilde, is a frugivorous bat which is uncommon but widely distributed in various forest ecosystems. It's categorized as an understory frugivore, yet this species is more commonly encountered in primary forest, whereas its close cousin, Stinera lilium, is more frequently captured in disturbed habitats. The diet of Stinera tilde mainly includes fruit, probably supplemented with insects. Fruit can be taken from trees such as the kiskadee, black kiskadee, and various fig species. This now brings us to the conclusion of this presentation on pollinators. Here is a list of our references used to conduct this survey. Please feel free to take a look at these references later. I would now like to thank everyone for taking time to attend this webinar today. And I would also like to thank our host at BES-Net TT for putting together this series of webinars. And special thanks to our group Trinibats for providing the equipment and support in conducting this survey on bat pollinators in northeastern Trinidad. Thank you all very much. Have a good day.
Okay, so welcome back, everyone. And thank you so much, Darshan, for that very interesting presentation, as well as the very lovely photos of the bats so that we can get a little bit more familiar with what they look like and perhaps how they are adapted to the particular type of feeding that they carry out. Okay, so we are now going to bring you on board live, Mr. Narang, so that we can move into our question and answer session. If you have any questions, members of our audience, please feel free to post your questions in our Q&A section of the Zoom platform. Okay, so I see we have a few questions, and I am going to read them out so that Mr. Narang can answer. Janine Sitahal has posted the Trinibats website, so we'll share that with you. M. Chin is asking, Darshan, what is the name of the reference text for identification?
Hi, Celeste, and hi everyone. I think we answered. So in the chat, sorry, in the Q&A section, you can see the answered question that we'll also type live during the presentation. I think Shane has included the field guide name. It's Bats of Trinidad and Tobago. Very simple, easy to find. I think it's on Amazon as well as locally in a few bookstores. And if you are a member of the Trinidad and Tobago Field Naturalist Club, you can get it there for a cheaper price.
Okay. I have another question, Darshan, really just a clarification. You mentioned that the mist nets were set up at ground level, and I know you gave the dimensions in your presentation, but could you just repeat how high up the mist nets go when you spread them across your research area?
That's a great question, Celeste. Yes, so it depends usually, right? It depends on the habitat that's been surveyed. So like with any experiment, you need to have your tools available, and it depends on the question we were asking and the location. So since we were serving two different habitats, one in the understory of primary / secondary forest, which is the Malao forest, and an open area in the quarry site, we have to standardize the types of mist nets. So mist nets come in all different sizes, heights, dimensions. And with these surveys, you basically, if you're doing this type of survey where you just want to get the question is what bat species are present and what of those species were pollinators. We use a standardized survey where the number of mist nets, when we calculated the mist net hours, that figure I gave there, 48 mist net hours, it's a standard effort number. So we can compare the effort from this project to other projects. The length of the mist nets vary widely from 3 meters, 6 meters, 9, 12, and 18. That is determined based on the length of the trail we are clearing, or if we are using an existing trail. The height of the single mist net is 2.7 meters, around 9-10 feet.
Mm-hmm.
If we have areas that is very open and we have accessibility, we can stack these nets 2 times up, 3 times up. So there is a standard net called a triple high, where it's a series of pulleys and flag poles, basically, where we stack 3 nets up. But that just depends on the habitat. And if we are doing understory work where we cannot set up triple high nets, we just use basic ground nets. So these are called ground nets. There's also one other type of net that's available. So different tools where if we were surveying the canopy, like upper canopy, someone might ask, what about the bats that are flying above the nets? Which preemptively, I will answer that we are surveying a different strata of the environment. So in a forest, you have the ground level, you have mid canopy, you have upper canopy. And ideally, we would want to set up multiple levels in a forest from the top of the canopy, which is based on what equipment is available. So for those types of more detailed surveys, part 2 hopefully, we can set up these canopy nets. Basically, we hoist a net up into the forest. Mm-hmm. Now, for the bats that are flying in the open air, those would not be recorded in these types of forest surveys, like the ones that fly way above the canopy in the sky at evening times. You usually see these bats zipping around, catching insects above the canopy. So usually we would survey those types of bats with acoustic meters, since we cannot really catch a bat 200 meters off the ground. So different tools for different questions. Right. Hope that answers the question.
Okay. There is another question. Kelly is asking, were you able to tell which species the bats may be pollinating by looking at the attached pollen or feces?
So yes, in this particular survey, because of the low numbers of individuals, the time of year that we caught the bats, we actually did not get any pollen on the species itself. If you're capturing bats when the flowers from trees are flowering, then you would more likely get pollen being deposited on bats. And usually this is around the dry season, March, April, May, when usually a lot of flowers well, a lot of trees are in bloom or flowering. So we surveyed this, we conducted the survey August, September last year. So we were not actually directly able to tell if any of our fruit-eating bats or nectar-feeding bats were actually directly pollinating species. So what we have in our list of species is what is known from the literature. These species were found there. So it was just a correlation at the moment of what are the likely bat pollinators in that area. So if we were to go back in the dry season, specifically when we identify what trees are flowering, we can go in, collect pollen from, so that is one level up basically in terms of the research, where you want to identify, tie down which direct species are being pollinated by which bats. Then you would go in, collect pollen from the faces or the body, the fur of the bats, and you would send that along to someone to identify this pollen. So for our survey, we were not able to see or observe any pollen on the bats.
Okay, and
Yeah.
yeah, and I think you alluded to it in your response, but I'm just asking again for clarification. The frugivorous and the nectivorous bats, the pollen would tend to be located perhaps on the head area. For those that are not necessarily frugivorous or nectivorous, you mentioned they may have incidental capture of pollen, and therefore, where would that pollen generally be found in those other types of bats?
Anywhere on the body, anywhere captured on the fur, because all of our bats here are, have some level of fur on the body, right? So by brushing past the flower, if it's feeding, as I mentioned, on insects, let's say there's a grasshopper landed on the flower and a bat flies by, picks it off. Maybe it might get it on its fur, on its face, different parts of the bat. With the fruit-eating bats, because they're moving around trees, grabbing fruits, you can get more facial features really. Not on the wings. I mean, the wings are not covered in fur; they are basically skin. So you will rarely get bats with pollen on the wings. They're usually trapped in the fur, and that's how it spreads from flower to flower.
Right. We have another question, Darshan. What were the common external parasites found on the bats?
Great question. Usually we get different species of bat mites and flies. So these are little yellow flies, little mites. I'm not we weren't particularly studying that question. So another survey you can do is to collect these flies and parasites, or total other specialty where you collect them in the field, transfer them to little vials of alcohol, and pass them on for analysis. But that is an area that needs a lot more work as well.
Okay. I've just posted in the chat something that Mr. Gomes shared in the Q&A, which is very useful regarding the availability of the publication Bats of Trinidad and Tobago. So everyone can check the chat to get that information. Moving on to another question from Jalaluddin. Can you comment on the site location, bat habitat, land management activities impacting on these protected species, and how can they be protected? Let me read that again. Can you comment on the site location, bat habitat, land management activities impacting on these protected species, and how can they be protected?
Thanks for that question, Jal. And yes, that's a great question, because these two study sites that we surveyed for this particular project tied into other BES-Net activities in the area. So we didn't select these randomly. These two sites, we were looking at the flora species as well. I think Dr. Floyd Homer had done work in this area. So we're able to correlate some of the conservation activities in the area. The Malau Forest Reserve is technically protected to some extent by the Forestry Division because it's listed as a forest reserve. So whatever management activities is happening there can show, if we are able to continue documenting the bat species over time, then yes, we can say there's a decline and increase in bat species there. But as far as I know, this was the first official survey, even though preliminary and very rapid survey of these two sites. So it's a baseline survey we conducted. The site at the Trinidad, the quarry site, that's recently rehabilitated site. So the acacia have only been about, when we were surveying our site, the height of the trees were less than 30 feet. So not fully rehabilitated sites on the early stages of pioneer species out there. So it's a good baseline in terms of future management and conservation of a rehab site and comparing it to a protected area. Ideally, we would also be targeting other sites in the area to compare pollinators in a, let's say, for example, in Matura National Park or Aripo Savannas, for example.
Okay, thanks very much, Darshan. There's a question from Patrick. Have you ever found ticks on bats?
Actually, not really. No, not any ticks to my knowledge on the individuals that we've handled. Mainly bat flies, bat mites, and this is just from observing the bats with our naked eyes, right? That's not my specialty in terms of looking at the parasites, but we have worked along with other researchers who have collected, parasites, what are known as the ectoparasites, parasites from the skin and the body. And a few other researchers would have collected endoparasites, looking at, taking individual bats and looking at parasites internally. But as far as I know, I don't know of any ticks locally on bats. But I would like to shout out Jeffrey Gomes, the author of the Bats of Trinidad and Tobago, as well as the founder of our group, Trinibats. So most of our knowledge comes from the pioneers in the field, such as Jeffrey Gomes, Luke Rostand, Janine Sita-Howe, just to mention the leaders in the bat research world.
Okay, thanks for that. And of course, we acknowledge Mr. Gomes' pioneering and sterling and continuous work in this area, which brings me to some of the data that was shared, Darshan. And you mentioned, according to the document that Mr. Gomes has put out 2015, which would be a few years back, some of the species that were found, and you indicated there were 19 species of our bats documented in this research. Some of the species under Mr. Gomes' listing were uncommon or rare, and you indicated that greater sampling effort or continuous sampling effort may in fact show these to be more common. I wanted to find out, of the 69, is that the correct figure? 69 local species that we have, are any of them listed on the IUCN listing as rare, threatened, or endangered?
No, it's not. There are no species listed as critically endangered, but that may be due to deficiency in the data. So with the IUCN Red List, that's a global list. So they look at things from a global perspective, a species level perspective. Now, when you bring it down to an island or a pair of islands off the coast of a continent, we would have population level threats. So it doesn't match up exactly with what the IUCN has for the global level. So our species that are rare locally are not necessarily what would be on that list, but we consider them rare, uncommon, and at the risk of being extirpated or removed from just our islands.
Mhm, mhm. Okay. I think there's another question in the, no. Actually, Mr. Gomes gave a correction. There are actually 70 species currently recorded locally. So thank you for that information, Mr. Gomes.
And I'm sure
Are there any?
would publish that in the second edition of the Bats of Trinidad and Tobago.
Yes. Okay. I wanted to ask one more question, Darshan, since there are no more in the Q&A. What is your recommendation for increased sampling of or increased surveying of our bat species locally? How frequently should it be done, and perhaps by whom?
Great question. I've asked myself and many other people this question, because it's an area of local biodiversity research or wildlife ecology research that requires different levels of expertise. So to handle bats, to get mist nets up is very specialized. So you really don't want every and anyone conducting that level of handling, right? There are other levels of research that can be done by anyone. And we are looking at a more holistic tools and equipment such as acoustic monitoring, which is the main field I would recommend if anyone wants to get into bat research, even bat observations, is to get handheld meters. They have their devices now that are small attachments, small microphones that plug into your Android phones, your Apple phones, that you can observe bat recordings, bat acoustics. So for the future, research-wise, in terms of getting more data, the cheapest and most available tools are with the acoustic monitoring. So for a round two project, for example, we would deploy remote acoustic monitors or samplers. Most people know about these for like whales and what's known as passive acoustic monitoring, or for birds, where you put out these microphones with recorders in the forest and they constantly monitor bat species. The caveat to that with acoustic monitoring is our library of calls. We need to build, and not just we, but I mean globally, on a database of these calls. With birds, you can hear the calls, you can identify them by ear. With a bat, because we cannot hear bats in terms of their acoustics, we need devices, we need equipment to deploy, which there's a cost to that, there's a cost of training, maintenance, etc. And then the skill and the analysis. So all of these methods, tools have pros and cons to deploying in the field. So one may ask, why isn't like someone like the forestry division or a protected area manager conducting his research? But the main reason is that it's highly specialized, highly technical in terms of monitoring and conducting research on bats.
Okay, thank you for that, Darshan. And we know that Trinidad has been doing sterling work, doing surveys, apart from what was done by yourself and a small team on this project. In our audience, I am quite aware that we have persons who are part of the Veterinary Students Association. We also have persons from Nurture Nature, and we have persons from the Ministry of Agriculture. I know that there is a bat monitoring or rabies monitoring that takes place within that. So perhaps there's opportunity for some collaboration, some collaborative work, or some collaborative brainstorming, perhaps, on what should be the best type of monitoring protocol to be implemented. for monitoring our bat species, a very important aspect of our biodiversity. So with that, I am keeping an eye on the time, and I think we can close off on our questions for right now. I take the opportunity right now on behalf of the BES-Net TT project team, therefore, to thank you, Darshan, Mr. Narang, and your research team for your work on this project. The BES-Net TT team is very much interested, as I indicated, in sharing the results of our research under the project, and we are hoping to have a publication come out of this research, and as well a report to be put onto our webpage. I'd like to thank you, the members of the audience, for your interest and participation in this webinar and your wonderful questions. Please remember that a recording of the webinar, as well as responses to any unanswered questions, will be posted to the project's Facebook page. I also wish to let you know that our webinar reporting on the research investigation of the seagrass flowering and pollination will be taking place next week, Wednesday, at the same time, 1 to 2 p.m. Please visit our Facebook and Instagram pages to obtain webinar registration information, as well as information on our upcoming project activities. We look forward to your continued interest and engagement in the BES-Net TT project. Have a good afternoon and a great day. Goodbye.